The Honorable Board of Examiners, Associate Professor Dr. Chandran and Dr. Kadil, the Honorable My Thesis Supervisor, Associate Professor Dr. Dan Yisin, thank you for giving me the opportunity to present my undergraduate thesis entitled Molecular Variation of Trichoderma Species Associated with Greenmore Contamination of Oyster Mushroom using Intersimple Sequence Repeat Marker or ISSR. My name is Go Mei Rong, and today I will talk to you on these 4 aspects of my project. Let me start by giving you some background information regarding the topic. Oyster mushroom is a basidiomycete that provides nutrients as an edible mushroom. However, the artificial cultivation has been seriously affected by a microfungi known as Trichoderma, and this has incurred a huge economic loss in mushroom industry. We can easily identify genus Trichoderma with its finger-like projections containing hyphae branched with conidiophores and phyalides. In early 90s, people had started to delimit the Trichoderma species using several of the earliest tools. Nonetheless, I do want to highlight the low reproducibility in these traditional techniques. According to the publication analysis, India is the country which reported the highest number of cases, followed by most of the European countries. To the best of our knowledge, not much studies on trichoderma delimitation have been reported in Asian countries up until today. Hence, the diversity of trichoderma in Malaysia remains obscure, and this makes it hard to identify the cryptid species of contaminants. As regards the issue, it is of interest to us. How can we differentiate these cryptid species of Trichoderma contaminants, which is important for designing the proper treatment? To answer the research question, our study aims first to review the current techniques available for Trichoderma delimitation, and second, to study the genetic diversity of Trichoderma species using ISSR. Before experiment, we hypothesized that ISSR is able to differentiate cryptid Trichoderma species. To fulfill the objective of research, I started off my project in analyzing the relevant journals for qualitative study. For quantitative study, I have cultured and observed 18 isolates before putting them into DNA extraction. Later on, I conducted ISSR PCR and gel electrophoresis to obtain the bands for trans-illuminator viewing. Only clear and distinguishable bands were accounted for similarity analysis and phylogenetic grouping. We spoke about 2 research objectives earlier, and let's now look at their results and discussion in more depth. First, we will talk about the current techniques in trichoderma delimitation. Findings show that additional morphological features have been used as preliminary identification in trichoderma species. The cultural morphology could be manipulated by the choice of media, and this may explain why we have to combine a number of morphological features to delimit the closely related trichoderma species. Moving on to the next finding, I have also found a few biochemical techniques which are useful to a certain degree in trichoderma delimitation. Among all, lipid phenotyping is feasible for differentiating closely related trichoderma, as each species will have distinct and conserved cell membrane lipid composition. Moving forward, our findings in molecular characterization had shown the gradual replacement of traditional methods by alternative techniques, for example FYP, SSR, and ISSR, while ITS remained as the most used technique for 20 years. Meanwhile, novel markers such as SCAR, CAPS, and SRAP have also been developed from the RFLP and RAPD techniques. Researchers have also suggested to use mitochondrial, ribosomal, or plasmid DNA with smaller genome size and higher mutation rate than nuclear DNA to differentiate the closely related trichoderma species. It is important to select the most suitable technique for study using several criteria as shown. After going through the first objective, I would now like to draw your attention to second objective focusing on genetic diversity of 18 Trichoderma species using ISSR marker. These are the 18 isolates belong to 9 different Trichoderma species in my study. Most of the Trichoderma cultures have small green or white conidia appearing in concentric ring, except for T. rhizae which appear in yellow pigmentation. In terms of micromorphology, all 9 Trichoderma species show an acute to right angle primary conidiophore branch with bowling pin phyalites. All 9 species in our study did not show secondary conidiophore as reported in Samuels. The species morphology concur with previous studies except for T. rhizae, which has no literature support. This leads to my next point on polymorphism analysis of Trichoderma strands. The 7 ISSR primer gave rise to 100% polymorphism with a total of 88 polymorphic bands. The study only included amplification products of 400 to 1500 base pair to avoid primer-dimer and weak intensity bands. Looking closely to the figure, you will notice that primer 15 yielded the highest number of bands and the least was in primer 10. From this finding, we can say that ISSR gave an average of 13 loci per primer. Meanwhile, our study had also reduced the self-annealing of primers by using specific dinucleotide motifs. It is important to also reduce the loading volume of PCR product to prevent band smelling, which makes scoring difficult and inconsistent. After doing similarity analysis for ISSR profiles, we obtained a genetic coefficient ranging from 0.3636 to 1. The lowest similarity was found in between Trichoderma puratum and Trichoderma lentiforme, while the highest similarity was recorded within these 3 pairs of Trichoderma strands. High similarity could simply mean the same origin or the need for more primers in strand differentiation. Using the similarity index, an APMA dendrogram was generated showing 4 large groups. The biggest group 1 was dominated with 7 Trichoderma puratum and 1 Trichoderma puraticola, and the second largest group 3 was occupied with class 2 of Trichoderma simonsi. It is surprising to see the genetic relateness between Trichoderma puratum and Trichoderma puraticola despite of their large phenetic divergence. Looking deeper into group 2, we can see 2 different species Trichoderma lentiforme and Trichoderma revi within the same cluster. A popular explanation for this is the identical ITS sequence and the same origin as reported by Chevery. Besides, the 2 different species Trichoderma rhizae and Trichoderma coningopsis were located in less complex group 2 as a result of revolutionary or random effects. Only Trichoderma asperorum in group 4 is distantly separated from the other strands. Overall, Trichoderma isolates have shown a high genetic diversity, and this is essential for the adaptation, survival, and infection. Furthermore, let me use the principal coordinate analysis to show you the spatial representation of the genetic diversity. The 3D plot accounted for 69.4% of the total variance observed in genotype, and the result of coordinate clusters corresponded with the grouping pattern in APMA dendrogram. Our data shows that species collected from different sources were clustered in the same coordinate, and this finding agreed with Melitor, who mentioned that geographical distance in mushroom farm could not act as a border to prevent gene exchange. Hence, it is plausible for Trichoderma species to migrate from one contaminated source to another, and this gene flow has actually contributed to the genetic diversity in Trichoderma population. In conclusion, our study had answered the research question by giving examples of morphological, biochemical, and molecular delimitation techniques. Besides, our result had also distinguished the cryptid Trichoderma species into 4 big groups and small clusters using 7 primers with 100% polymorphism. In future, we suggest to use ISSR analysis in determining site-specific remote pathogens. That's all from me, and thank you for your attention.